So on this picture right here, of course, you still see your DNA. Agreed? And then let's just say that this is the 5' end. So this would be the 3', this would be 3', and this would be 5' end. Okay. What you have in this little section right through here is your TA area. TA box. So just repeating units of TA and TA. And let's say that this represents right here the start point of transcription. What has to happen, and you see one of these guys here, is you have to, or the only way, I should say, the only way to start this process of transcribing DNA into RNA is the binding of different transcription factors. And here you've drawn the first binding to this region to help promote the binding of RNA polymerase. So I could continue to draw in more of these transcription factors. So every single circle that I've drawn here is representing a transcription factor. So I'm showing you by the arrows here that everything's a transcription factor. All we need is a bunch of circles around this box area. Now, this makes a lot of sense, I think, why you would need so many different transcription factors. Can anyone like suggest or theorize why you would need not just one transcription factor, but more like 20 to go ahead and do this process of making some RNA? Could be catalyze. I think it's something a little bit more involved. Usually when big decisions are made, is it just one person, or do you have like a team of cohorts or a team of collaborators working together to come up to decide one outcome? It's a team, right? In other words, you're not going to do this transcription process unless you can guarantee that everything that you need is set to go, right? So I want to make sure we can see that you got to have all of these units present in order to just bind the RNA polymerase. Now once they're all there, your RNA polymerase enzyme, which is going to be huge, can bind. So this guy that I've just drawn in is RNA polymerase. And once it's there, you can start to go ahead and read the DNA, right? And you'll see in this drawing, which you don't have to draw, RNA polymerase has started to read the DNA. It will continue to synthesize a chain of RNA nucleotides. This would have to be 5', this would have to be 3', which would make sense because we'd already labeled this as 5 and 3 right there. Right? And it will synthesize this RNA strand until it reaches a terminating sequence upstream, as we say. Good? As far as the process or step 2 of this, because we have just talked about how you can initiate it, you have to have transcription factors, as you saw from the drawing, bind, and then RNA polymerase can bind. Step 2 is going to be about elongating the strand. Again, we're still talking about steps of transcription. To elongate the strand, of course, this is going to be the work of RNA polymerase. It can untwist the DNA just enough to copy it or transcribe it. This process is at a rate, so RNA polymerase can add about 40 nucleotides per second. And the other unique thing to mention as well is that you can have multiple RNA polymerase enzymes reading a single sequence of DNA. Which means you can make a whole lot of these mRNA transcripts in a very short period of time. Good. And then of course our last step here is going to be step 3, which is the process of termination. And it's pretty simple. Termination is going to occur when RNA polymerase comes to the terminating region. Actually just drops off, and that's all that's left. Does RNA polymerase destroyed? That just means the region where everything stops. Stops at the terminator region. So it's not destroyed. To terminate something means to stop it. Doesn't mean to kill it. Too many movies, not that good. Right? What's after transcription? Okay, kind of.
It's RNA splicing.
RNA splicing. Now this is probably going to be something that we would see in eukaryotes mostly. So this process is found in eukaryotic cells, and that's of course because they have a nucleus, right? And we're going to alter a lot of things. One of the first things we're going to do is we add, or we don't, but inside the nucleus there is an RNA molecule that's transcribed, and we add to that a 5' cap. On the 5' end. We also take the 3' end and put a poly-A tail on it. All that means is that we put a bunch of adenine RNA nucleotides on it. I'll let you draw this little guy real quick. Real simple. You'll notice on this drawing, again, I could label this as maybe the 5' end, and this would be the 3' end. Here's your cap and your tail. And you'll notice that the darker portions are what is down here, and you see the arrow, so this must be like a before and after. Right? And before, you have something called pre-mRNA. That's what this is, pre-mRNA. RNA that still the RNA that still needs to be modified, and this would be real mRNA. Again, you'll notice that there's still that 5' cap on it. That hasn't gone away. You also see the poly A tail on the 3' end. But again, you'll notice that you've got just this darkened portion, and the light portions right here are missing. And we'll pick up on this tomorrow, but the section that's right here, that's the light, is going to be, that's an intron right there, and that's an intron right there. A segment that is removed or excised out, right? And these pieces that are still left in there at the end are called exons. So there's an exon there, an exon's there, an exon there. And remember that it's exons that end up in the final mRNA. And of course this mRNA then will exit the nucleus. Right? And it will be translated later on.